In clinical psychiatry, exposure therapy is a highly effective treatment for severe trauma, but it requires the patient to consciously recall and confront the exact memory causing their distress. For many, this process is so excruciating that they drop out of treatment entirely. This creates a significant failure rate for those suffering from post-traumatic stress disorder. Decades after returning home, veterans and trauma survivors are often left with treatment-resistant conditions, unable to bypass the pain of their own memories. The difficulty in isolating and treating a single traumatic memory lies in the physical nature of the brain. The cortex is a high-noise environment, cascading with millions of erratic electrical impulses every second. Conventional neurofeedback attempts to regulate this environment using a univariate approach. It reads the average electrical signal across a broad, general brain region, allowing the patient to practice upregulating or downregulating that overall area. While this can help induce generalized states like relaxation, the spatial resolution is extremely low. Averaging the signal of an entire lobe cannot isolate the highly specific, localized neural representation of a single memory or fear. To target a specific thought, we need an entirely different level of precision. This is where Decoded Neurofeedback, or DecNef, enters the picture. Instead of reading broad regional averages, DecNef uses functional magnetic resonance imaging paired with multivariate pattern analysis. This allows researchers to track complex, multidimensional voxel patterns deep within structures like the superior temporal sulcus. By shifting to this multivariate tracking, clinicians gain the precision necessary to identify the exact neural fingerprint of a specific trauma. They can then surgically edit that representation, reducing the physiological fear response without the patient ever consciously experiencing the memory. Before DecNef can alter a traumatic representation, researchers must map its exact coordinates in the fMRI scanner. They need to build a decoder. This introduces an immediate clinical paradox. To build a decoder that recognizes a traumatic stimulus, the patient must face that trigger inside the scanner. This causes the exact distress the therapy is meant to avoid. Early methods bypassed this using continuous flash suppression. By presenting the feared image to one eye and a rapidly flashing high-contrast pattern to the other, the visual processing is suppressed, rendering the presentation subjectively less distressing to the patient. A more advanced workaround removes the need to present aversive stimuli entirely. This technique is called hyperalignment. Hyperalignment maps a common representational space using data from surrogate participants viewing the feared object. By aligning these healthy neural responses, the specific target decoder for the traumatized patient can be mathematically inferred. Once the target data is acquired, the raw blood oxygen level dependent, or BOLD, signals must be computationally filtered. The system analyzes the data to isolate only the voxels that reliably classify the specific fear response. To achieve this, researchers apply sparse logistic regression. This algorithm automatically discards irrelevant brain noise and locks onto the exact voxels that carry the target information. Resolving this measurement paradox yields a precise target map of a trauma. Clinicians now have the exact coordinates to target, and they acquired them without re-traumatizing the patient. With the target decoder built, the patient enters the fMRI scanner for the induction phase. The instruction they receive is deliberately vague. They are told only to somehow regulate their brain activity to maximize the size of a visual disc on the screen. This setup initiates the DecNef closed-loop system. Data flows continuously from the patient's brain into the computational decoder and back to the patient as visual feedback. During a trial window, the patient spontaneously generates a multivoxel pattern in their brain. Because fMRI measures blood flow rather than instantaneous electrical spikes, the system must account for a 6-second hemodynamic delay between the actual brain activity and the scanner's measurement. Once the signal arrives, the decoder overlays the patient's incoming pattern against the pre-recorded traumatic target map to calculate a percentage of similarity. That calculated match translates directly into the size of the visual feedback disc presented to the patient. The larger the disc, the higher the financial reward. The scoring metric is strictly based on the mathematical likelihood that the patient's induced pattern matches the target pattern. Throughout this entire process, the patient has absolutely no conscious knowledge of what specific thought or memory they are being rewarded for activating. Through blind trial and error, the unconscious mind searches for the rewarding state. Eventually, it hits the exact neural configuration of the targeted fear. When that pattern aligns, the monetary reward reinforces the neural pathway, encouraging the brain to recreate that specific representation in subsequent trials. By repeating this closed loop, the system tricks the brain into repeatedly activating a highly specific traumatic representation, entirely below the threshold of conscious awareness. Data shows that DecNef objectively reduces physiological fear responses, but identifying the exact learning mechanism driving that reduction requires a closer look at the trial data. There are two competing theories for why the fear subsides. The first is exposure-based extinction, where safe activation diminishes its power. The second is counterconditioning, where reward actively overwrites the fear. This equation represents the Rescorla-Wagner model of classical conditioning. Researchers applied this mathematical framework to separate the two effects and isolate the variables driving the behavioral change. The exposure-based effect is modeled as a linear function. Its impact is strictly proportional to the number of times the target neural pattern is successfully induced. Conversely, the counterconditioning effect is quantified by multiplying the likelihood of the target pattern by the magnitude of the financial reward earned in that specific trial. When researchers analyzed the mixed effect model, the predicted values mapped to the experimental amygdala reactivity. The negative coefficient of the exposure-based effect emerged as the dominant statistical driver. The math points to a distinct clinical reality. DecNef achieves the gold standard extinction learning of traditional exposure therapy, entirely bypassing the conscious psychological distress of reliving the trauma. Beyond clinical psychiatry, cognitive psychologists have used DecNef to test the absolute precision limits of representational editing. A study published in Nature Communications posed a central theoretical question: can DecNef surgically edit a subject's metacognitive confidence in a decision without altering their actual perceptual accuracy? To test this, researchers used a dot motion discrimination task. Subjects had to decide which direction a cluster of dots was moving, separating their raw sensory perception from their internal confidence rating. This graph shows a linear discriminant function, mapping the fMRI data to prove that confidence isn't merely a byproduct of strong visual evidence. First, it plots the perceptual data. It separates the correct visual judgments from the incorrect ones based on signals in the visual cortex. Next, it plots the metacognitive data, establishing the boundaries for high and low confidence states based on voxel patterns in the frontoparietal network. Through a process called rectification analysis, researchers demonstrated a definitive boundary shift. The spatial activation patterns for confidence are structurally distinct from the raw sensory signals in the visual cortex. They do not perfectly overlap. With distinct targets isolated, the DecNef training intervention began. Researchers actively reinforced either the high or low confidence voxel patterns in separate groups. The neurofeedback successfully forced bidirectional changes. Subjects reported higher or lower confidence strictly corresponding to the specific pattern they were trained to induce. But the most important metric was the control variable tracked throughout these confidence shifts. Throughout the entire manipulation, the subject's actual perceptual accuracy remained completely unchanged, holding steady at exactly 75%. This empirical data proves subjective confidence and perceptual evidence are processed through entirely dissociable neural mechanisms. The brain is a dense, noisy architecture, but we now have the mathematics to isolate its exact components. Both the trauma studies and the metacognitive trials demonstrate that complex, high-order mental states are encoded in distinct voxel patterns. Once identified, these patterns can be isolated from the surrounding noise and directly manipulated. In clinical psychiatry, this translates to a viable, distress-free pathway for patients who cannot tolerate conventional exposure therapy. In computational neuroscience, it provides a causal tool to cleanly uncouple neural variables that traditionally overlap, allowing researchers to study the mechanics of thought with precision. By measuring at the multivariate level, we can map the coordinates of fear, confidence, and memory, tracing them directly to measurable behavioral outcomes. DecNef transforms neuromodulation from a blunt instrument into a highly targeted surgical tool, upgrading how we can heal and understand the human mind.